So this is my midterm video project. So the first thing I'll be discussing are lab practical skills. So an example of this, to better explain it, I thought it'd be better to use an example. So this is an example of simple dilution. So as you can see here, this is a 50%. So it starts off with 15 and you basically divide it each time by 2 or multiply it by 0.5. You're taking half of it and putting it into the next jar, and then you're also adding in 20 centimeters cubed of the water. And at the very end, there's a total of 40 centimeters cubed because of the ethanol, the 20 centimeters cubed of ethanol and the 20 centimeters cubed of the water. This is a single line graph, and the important thing to remember is that the IV is always going to be on the x-axis and the dV is always going to be on the y-axis. And just basically you copy the same exact labels that it has here, and then using a straight line, or using a ruler, you have to make straight lines, and then it's always important to either circle your points or use an X. This is an example of a bar graph, so very similarly, use a ruler to make straight lines. You put your labels on the x-axis and then your values on the y-axis, and then find a scale that's appropriate. This is a simple dilution. So to do this, you have to use the C1 V1 equals C2 V2. So the C1 is the initial concentration, and that's always going to be given. The V1 is what you're basically trying to find. The C2 are the concentrations that you need to make, and then the V2 is how much of concentrations that you need to prepare, and that's also always going to be given. In this case, it is 10 centimeters cubed. So on here is a C2. So basically using this formula, and you're just basically plugging everything into there. So that's how I got my values for the volume of the hydrochloric acid. And to find the volume of distilled water, I basically took, since V2, to find that, it's the V1 plus the W. So I took V2, which is the 10, and I subtracted by the V1 to find the amount of water here. So next up are independent and dependent variables. The independent variable are the factors or values that you decide on and change. And the dependent variables, you can't control those, so that's what you measure and collect your data with. So measuring the dependent variable, so sometimes they're going to tell you how to do this, but sometimes you have to decide yourself. So you can either determine the initial rate of reaction, or you can decide, or you can use the method of how to take the endpoint, which is the identifiable stage in the reaction. So you have both quantitative, which is a numerical result, and qualitative data, which is the data that is not numerical. So you can use a colorimeter, which measures the amount of light that is absorbed by a tube that contains a color liquid. So the deeper the color means the more light is absorbed. And it's helpful to use color standards to compare color, so you can get more accurate and precise results. So when making conclusions, it's important to have a simple and well-focused clear statement, and then it should also be related to your first question or your hypothesis. When describing data, it's best to show the graph that you created. So you want to show the trends and the patterns between the independent and the dependent variable. And then it's very also important to use numerical data from the graph. So when identifying sources of error, there are two types, which is systematic and random error. Systematic are errors that are likely to be the same throughout the experiment, and random errors are the errors that are different for different stages of your investigation. When suggesting improvement, it's important to use techniques that when measuring the dependent variable that are more reliable, so such as the colorimeter, or using controlled variables. So for properties of the water has a lot of properties, and I'm just going to go through them. So the first one is that water is a universal solvent. Water molecules are attracted to ions of polar molecules. They collect, surround, and separate them. So this occurs in the body when a chemical dissolves in water. So the next one is water is a transport medium. So in animals, it serves as a transport medium in blood, the lymphatic, the excretory, and the digestive system. In plants, it serves as the vascular tissue. So what is specific heat capacity? So it's the amount of heat that's required to raise a substance's temperature by a given amount. So water-specific heat capacity is the amount of energy needed to raise the temperature of 1 kilogram of water by 1 degree Celsius. So water has an extremely high heat capacity because of its hydrogen bonds. It needs more energy to move around to break the bonds between them. So this is important because water is more resistant to temperature changes. So within the body, it becomes more constant. There's not a lot of difference. And then in large bodies of water, it becomes a more stable habitat for organisms, especially for the aquatic ones. And then in high latent heat of vaporization, it's the amount of heat needed to vaporize a liquid. So in this case, it'd be turning from liquid water to water vapor. So because of water's high heat capacity, it has a high latent heat of vaporization. The water molecules stick together, so it needs a lot of energy to basically break the bonds, the hydrogen bonds, and vaporize the water. So for density and freezing properties, water is less dense in its solid form, and because of this, ice floats on liquid water and insulates the water under it, and it increases the chances of life. And then density changes also with temperature, which helps to maintain the circulation of nutrients because of the currents. So it also has high surface tension and cohesion. So such as these pond skaters here, it's able to stand on it and use it as the water as a habitat. So it has high cohesion, which means that the molecules basically stick together and can move through unbroken columns through vascular tissues and plants. Next up is biological molecules. So the building blocks of life are macromolecules, polymers, and monomers. So first up is carbohydrates. So these are basically the first type is monosaccharides, which are basically sugars, starting from stage 2O, and it's only one sugar molecule. This is the structural formula, and this is the ring structure. So in alpha glucose, the hydroxyl group is below the hydrogen, and in beta glucose, it's above the hydrogen. So disaccharides are two monosaccharides that are joined together. They are still sugars. And basically how disaccharides are created is through condensation reactions. So it's when two hydroxyl groups line up, and then basically one water molecule is formed, means like taken out, and it forms a glycosidic bond. Hydrolysis is the opposite reaction needed to break down the glycosidic bond and then just make them back into monosaccharides. So polysaccharides are polymers, which their subunits are monosaccharides, and they're made by joining together monosaccharides through condensation, and they are not sugars. So the first type is starches, which is found in plants. The starch is made from amylose and amylopectin. So an amylose is made from alpha glucose. They're made from unbranching chains, and they coil into helical structures. Amylopectin is also made from aglucose, and they're made from 1,4 carbon chains and also 1,6 carbon chains, which means that they also branch out. Glycogen is found in animals. It's very similar to amylopectin. It contains 1,4 and 1,6 carbon chains, meaning that they are branched, and it's also composed of alpha glucose. Cellulose is made from beta glucose, and they're linked together at 180 because of this. Hydrogen, they're made from hydrogen bonds, which are individually weak, but they provide enormous strength when joined together, and this helps to withstand the pressures of osmosis. Next up is lipids, which are the organic molecules that are insoluble in water. So the fatty acids are a type. They contain the carboxyl group, and the tails may have double bonds, which are unsaturated here, as you can see, and then no double bonds, which is saturated. So next are the alcohols and the esters. So alcohols are a series of organic molecules with a hydroxyl group, and the reaction between the acid and the alcohol creates an ester, and this is the chemical link is all referred to the ester bond. This is a triglyceride where there's 3 fatty acid tails and 3 ester bonds. They are insoluble in water, meaning that they are hydrophobic. So functions of triglycerides is that it serves as an energy reserve. It has a higher energy yield than carbohydrates. It serves as an insulator, and in humans it's stored below the dermis and around the kidneys. It's an insulator against heat, as it's for blubber, and it's also a metabolic source of water, as you can see with this right here. These are the phospholipids. So one of the 3 fatty acid tails is replaced by the phosphate head here. So it means that it's hydrophilic, it has a hydrophilic head and hydrophobic tails. So next up is proteins. So proteins are made from amino acids. They have a central carbon, as you can see here, that is bonded to an amine group and is bonded to a carboxylic acid group. And there's also hydrogen. The only way, as you can see, they're all similar except for their R group. So they're joined together, very similar to the glycosidic bond, but instead it's a peptide bond. Similarly, they have a condensation reaction, which is basically a H2O molecule is taken out, and it can be broken up through hydrolysis. So in the primary structure, it's the sequence of amino acids in a polypeptide chain, and the secondary structure, it consists of the coiling and folding of a chain of amino acids through hydrogen bonding. So as you can see here, this is the alpha helix, and this is the beta glued sheet. Next up is the tertiary structure, and is the way in the protein coils up in a precise 3D shape. So this includes the hydrogen, disulfide ionic, and the weak hydrophobic interactions. And then here's a 3D arrangement of 2 or more polypeptides, which is referred to as the quaternary structure. So this is just a model of the tertiary and the quaternary, and you can see all the different chains there. Next up are globular and fibrous proteins. So globular proteins are basically proteins which curl into a ball shape. and then fibrous proteins, which form long strands, but they're not soluble in water, but they do have structural roles. So an example of a globular protein is hemoglobin. It has 4 polypeptide chains, 2 of which are A chains, which are made from alpha globin, and then the other 2 are beta chains, which are made from beta globin. It contains a heme group, which is also referred to as peptide group, and is not made from amino acids. So this is collagen, which is an example of protein. So it's important structure of protein in animals. It has 3 polypeptide chains, as you can see here, in the shape of a helix. Every third is glycine, which is the smallest amino acid, and interacts with other collagen molecules. It's very flexible and can withstand tensile strength. Next up are chemical reactions and enzymes. So what are enzymes? Enzymes are biological catalysts. They're globular proteins, which means that they're water soluble. They're unchanged after the reactions. Nearly all metabolic reactions happen because of enzymes, and they help to reduce activation energy. So there's intracellular and extracellular. So intracellular basically inside the cell, and then extracellular is the ones that are secreted out of the cell. So the fit and key, the lock and key induced fit in the hypothesis. So basically the lock and key theory basically says that there's a cleft and depression, which is the active site, as you can see here. And the active site and the substrate has a complementary shape. The substrate meets the enzyme and it fits into the cleft. The R group binds with the substrate and the complex is formed, and the enzyme catalyzes the reaction. Product is formed and leaves the enzyme, and the enzyme is like left completely fine. The induced fit theory basically says that it's the same exact thing as the lock and key theory, but it's more flexible, but it can slightly change shape so it can better fit the substrate. So next up is the factors on the rate of enzyme catalyzed reactions. So pH, which can disturb the ionic bonds and change the charges of amino acids. You can also increases in temperature, which makes the molecular movement speed up. There's more random movement, which means more reactions. The optimum temperature is the temperature where enzymes catalyze at its maximum rate. And if it's too high, the hydrogen bond breaks, which means that it denatures the enzyme. And decreases in temperature results to less active enzymes, but it does not denature the enzymes. Factors on the rate of enzyme part 2. So substrate concentration. So as the substrate concentration increases, the initial rate increases as well. And then in enzyme concentration, there's a large at the beginning of the reaction, there's a large number of substrate molecules. Every enzyme has a substrate in its active site, but then as more and more substrate is converted, there's less substrates available. So then the reaction slows down until it completely stops. So when comparing enzyme affinities, the higher the enzyme affinity, the more likely the product will be formed as a lower Km value, which is you can see here. And then a nice comparison is that maximum speed and acceleration of the car. So next up are enzyme inhibitors. So they're competitive reversible inhibitors, basically prevent the substrate from joining the enzyme. The Km value increases, the Vmax doesn't change, but this can be reversed if the substrate concentration is increased. Non-competitive reversible inhibition changes the enzyme shapes, but because of this, it cannot, no product can be produced anymore. So when immobilizing enzymes, so it's basically used for commercial purposes. So it's basically, this is an example of this would be lactase when making lactose-free milk. So basically they form beads, as you can see here, they're arranged in a column and milk is poured through it. And then some advantages is that you do not need to separate the enzyme. The milk is not contaminated with the enzymes. The lactase is not lost, and it's also tolerant to pH and temperature changes in comparison. Next up is cell structure and function. So this is the cell theory, where it basically states that all living organisms are made from cells. Cells are the basic unit of all structure for all living organisms, and cells come from pre-existing cells. So animal and plant cells, what they have together they share similarly is that they are surrounded by the cell membrane, has a nucleus, it has cytoplasm, it has organelles, the mitochondria, and the Golgi apparatus. The differences is that animal cells contain centrioles, and then plant cells have a cell wall, central vacuoles, and chloroplasts. To calculate the size of a cell is that you need to take the observed size of the image and the actual size, and this is the magnification. Resolution is the ability to distinguish between two objects that are very close together, so it means that high resolution equals greater detail. So the types of microscopes are electrons, so transmission electron microscopes beam electrons that passes through the object, and scan electrons basically just scan the surface. So these are some of the organelles that are found in the eukaryotic. So nucleus, it holds all the DNA, ribosomes build proteins, The smooth ER builds the lipids and the fats, and then the rough ER folds the proteins. The Golgi body sends packages to areas outside the cell. The vacuole is storage, lysosome breaks down things. Mitochondria produces the energy, which is like the ATP stuff, and then the chloroplast helps with photosynthesis. So as you can see here, these are all the organelles and their labels, respectively. So the Indo-Cinderella theory basically says that mitochondria and chloroplasts are ancient bacteria that live inside the larger cells. So this provides support that eukaryotic cells were branched off from prokaryotic cells. So prokaryotes versus eukaryotes. So prokaryotes lack a nucleus, they're small in comparison to eukaryotes. There's no ER, and examples of this would be bacteria. Eukaryotes have a nucleus, there's an ER, there's many organelles, and examples would be plants, animals, and fungus. So viruses do not have a cell structure. They consist of self-replicating molecules such as DNA and RNA, and there's a protective coat of protein molecules. They're parasitic by reproducing, by infecting, and taking over living cells. And that is all.